Welcome back to the deep dive. Today we are taking a structured, really comprehensive plunge into the machinery that defines life itself.
DNA replication and the repair systems that guard our genetic blueprint.
Exactly. We are treating a deep technical source on molecular biology as our map, making sure we cover, every critical mechanism and every conceptual insight.
And it's such a fundamental mission. I mean, the stakes are, well, they're everything.
Right.
Every instance of reproduction, every single moment of a cell's life, it all hinges on the ability of DNA to be copied perfectly. But it's not just about copying. It's a constant war against decay.
That's a great way to put it. When we talk about maintaining genomic integrity, I think we tend to picture these big catastrophic events.
Sure, like radiation exposure.
But the source material really reminds us that damage is a constant, almost mundane reality.
Oh, absolutely.
Yeah.
We're talking about thousands of assaults every single day. The numbers are actually staggering. A typical warm-blooded mammal, it loses something like 10,000 bases from its DNA every day.
10,000 per day.
Per day. And not from anything exotic. Just ordinary environmental things like UV light from the sun or, chemical insults. And even just the byproducts of your own metabolism.
Wow. That scale of daily wear and tear, it really shows why survival isn't just about a good copier. You need an elite proactive maintenance crew.
And if that damage piles up, or if mistakes aren't fixed, they inevitably lead to mutations. And if those mutations hit the genes that regulate cell division, that cell is now squarely on the path toward cancer.
And we don't have to look far for, the real-world consequences of that. The BRCA1 gene is a really stark human example.
It is. And it's important to be clear what BRCA1 is. It's not a proto-oncogene. Its job, the protein it makes, is to be a crucial player in the highest fidelity DNA repair pathway we have.
The one that fixes double-strand breaks.
The most dangerous kind of damage. So if you inherit a faulty copy, faulty allele of this gene, you're compromising your cell's ability to do that really critical high-quality maintenance.
Right. Your maintenance crew is short-staffed.
Exactly. And so individuals who carry certain non-functional alleles of BRCA1, they face a substantially increased risk. We're talking 3 to 7 times higher risk of developing breast cancer, and often ovarian cancer too.
So let's unpack this a bit more. The ability to diagnose this risk genetically is a huge advantage, right? It's an incredible early warning system.
It is.
But it immediately forces a person to make these intensely personal, really difficult choices about prevention.
Agonizing choices. I mean, on one hand, you have the most aggressive option, which some people do choose, preventive surgery, like a double mastectomy.
Which is a massive intervention.
A massive intervention. But the studies show it can reduce the lifetime risk of breast cancer in carriers by around 90%. That is a life-changing number.
And what's the other path, the less invasive one?
That involves heightened, basically perpetual surveillance, increased screening, annual MRIs, frequent mammograms, all in the hope of catching any cancer that develops at the absolute earliest, most treatable stage.
The fact that one inherited defect in our molecular biology can force people into those kinds of high-stakes decisions. It's a very sobering place to start our deep dive.
It grounds the entire conversation. The incredible precision needed to copy the DNA in the first place, and then this 3-tiered defense system, the repair mechanisms that keep all that daily damage from just overwhelming the system.
So let's start at the absolute beginning, the foundation of life itself, the concept of duplication.
Right. The source material frames replication as fundamental at every level, organismal, cellular, genetic. But the history here is really important.
What's fascinating is how complicated the process is now compared to what we think existed in primordial life.
Exactly. Early genetic material was probably RNA. And RNA is simple enough, structurally, that it could have self-replicating capacity. But when life evolved to use DNA, which is, a much more stable, permanent archive, it lost that ability.
So DNA carries the blueprint for its own duplication, but it's chemically inert.
It needs the machinery. The source material has a great analogy. It says the image of DNA as self-replicating is like describing a letter as a self-replicating document. It's not. It needs a photocopier.
And the mechanism for that photocopier was suggested basically the moment the structure was revealed, back in 1953.
Yep. Watson and Crick proposed that because the 2 strands are complementary, and they're held together by those relatively weak hydrogen bonds, they could just separate, like a zipper.
And once they're separated, each of those parental strands could then serve as a template to direct the synthesis of a new complementary strand.
Sounds simple, but it actually generated 3 distinct conceptual models for how the final product would look, and they had to be tested.
Okay, so it's a question of distribution. What were the three hypothetical schemes?
The first, and the one that turned out to be the winner, was the semiconservative replication model. This is the one Watson and Crick's structure really predicted.
In that model, after one round of replication, each of the two daughter DNA duplexes would be a hybrid.
Exactly. One original parental strand and one brand new synthesized strand. So half of the original molecule is conserved in each copy.
Okay, that's model one. What was the second possibility?
The second was called conservative replication. This idea was that the two parental strands would somehow stick together after they were used as templates.
So you'd end up with one daughter molecule that's 100% old DNA and another that is 100% new DNA.
Precisely. The original is perfectly conserved as one unit.
And the third idea was a bit more complicated. Dispersive replication.
Yeah, this model suggested that the parental strands would be broken up into little fragments, and the new DNA would be synthesized in short bits in between. So the final strands would be this mosaic, a composite of old and new fragments all mixed together.
Why would anyone propose that? It sounds so messy.
Well, you have to remember why. Thinkers like Max Delbruck suggested it was maybe the only practical way to replicate a long, circular, intertwined molecule without having to unwind the whole thing.
Which seemed at the time like an impossibly complicated problem.
A topological nightmare. So how do you distinguish between these three ideas at the molecular level? The definitive proof came from that incredibly elegant 1957 Meselson Stahl experiment.
We were using bacteria, E. coli.
Right. They needed a way to label the parental DNA so they could tell it apart from the newly made DNA, and they used nitrogen isotopes to do it.
Heavy nitrogen, 15N, and the normal light nitrogen, 14N.
So first, they grew bacteria for many generations in a medium that only had heavy 15N, so all the DNA in those cells would be heavy.
Then they switched them. They moved the cells into a medium containing only the light 14N and let them replicate.
For 1 or 2 generations. And the key, the real genius, was how they separated the different densities of DNA.
This is the equilibrium density gradient centrifugation.
With cesium chloride, you spin this stuff at incredible speeds, and it forms a stable density gradient in the tube. DNA molecules will then float or sink until they hit the exact spot where their density matches the density of the solution.
So if replication was conservative, after one generation in that light medium, they should have seen 2 separate bands?
Exactly. One heavy band for the old DNA, and one light band for the new DNA.
But that's not what they saw.
Not at all. After one generation, they saw only a single band, and its density was exactly intermediate, a perfect hybrid, halfway between the heavy DNA and the light DNA.
So that result right there instantly eliminated the conservative model.
Gone. But it left semi-conservative and dispersive, since both could potentially produce a hybrid density molecule.
The tiebreaker came after the second generation.
Right. They let that hybrid DNA replicate again in the light medium, and then they saw 2 bands. One was still at that intermediate hybrid density.
And the other?
The other was a new band, entirely at the light density. And that distribution, 1 hybrid molecule for every 1 new light molecule, was the smoking gun. It unequivocally supported the semi-conservative model.
And it ruled out dispersive, which would have just produced a single band that was gradually getting lighter and lighter over time.
A smear, yeah. It was definitive.
Okay, now this is where it gets really fascinating, because the source material points out this fortuitous experimental artifact that was actually necessary for the experiment to even work so cleanly.
This is one of my favorite stories in science. The problem is that bacterial growth is asynchronous. Different cells start replicating at different times.
So if they had been able to measure the density of the whole intact circular bacterial chromosome,
the results would have been a mess. An intact genome gradually incorporates light nitrogen, so its density would just continuously shift over time.
it would have created a smear.
A smear that would have looked exactly like the prediction for the dispersive model. They might have drawn the wrong conclusion.
But because they inadvertently broke the huge genomic DNA into small fragments during the extraction
They
process,
essentially homogenized the sample. On that small fragment level, the fragments were either fully replicated or they haven't started yet. So breaking the DNA allowed them to see those clean, discrete density bands that define the semi-conservative pattern.
it was a lucky break that ensured the results were clear enough to define the mechanism for generations.
Absolutely. So that settled it in eukaryotes.
Yeah.
To prove it in eukaryotes, which have these huge linear chromosomes, we look to the Taylor experiment.
Which actually came a little earlier.
Right. Taylor used culture cells and a thymidine analog called bromodioxyuridine, or BRDU.
Which gets incorporated into the DNA.
Exactly. And then he used special stains to visualize the chromosomes. After one round of replication with BRDU, both chromatids of a chromosome were stained. But after 2 rounds, you could visually see the semiconservative pattern.
What did it look like?
One sister chromatid was fully substituted with BRDU, so it stained differently. And the other was a hybrid, one BRDU strand and one normal strand. It was visual proof that the same mechanism held true even for our own complex chromosomes.
Let's move our focus from the abstract concept to the actual physical machines that do the work. We're diving into the prokaryotic replication machinery.
And progress here was really driven by 2 key experimental strategies. The first was the use of temperature-sensitive mutants.
Or Thix mutants.
Right. Replication involves dozens of essential proteins. If you just knock one out completely, the cell dies and you can't study it.
But with a Tis mutant, the protein only fails at a high, non-permissive temperature.
So researchers could grow the cells at a normal permissive temperature and then crank up the heat and see exactly where the replication process breaks down. It allowed them to systematically figure out what all the genetic components were doing.
And the second crucial tool,
In vitro reconstitution systems, which basically means purifying every single component. And there are over 30 different proteins in E. coli, and then putting them all back together in a test tube to see what combination was the minimum required to get replication to happen.
So we know replication starts at a specific site on that circular bacterial chromosome.
The origin, or oryx.
And once it starts, it proceeds bidirectionally. It forms 2 replication forks that move in opposite directions around the circle until they meet on the other side.
And moving those 2 forks brings us right back to that massive physical challenge we mentioned earlier, the unwinding problem.
Right, the topological nightmare. You're trying to separate the 2 strands of this highly coiled circle.
And doing that introduces immense torsional stress. It's like trying to unwind a rope that's nailed down at the other end. You just get more and more knots and tangles ahead of where you're working.
In the DNA, that manifests as an accumulation of positive supercoils ahead of the fork.
Which would eventually just seize up the whole process. It would grind to a halt.
So how does the cell solve this impending disaster?
The solution is an amazing enzyme called DNA gyrase. It's a type of enzyme called a type 2 topoisomerase.
And the type 2 part is the key here.
It is. Type I topoisomerases usually just nick one strand. But type 2 enzymes, like gyrase, are much more powerful. They cleave both strands of the DNA duplex.
They cut the rope completely.
They cut the rope, pass another segment of DNA through the resulting break, and then seal the strands back up again. It's an ATP-driven process.
And that double-strand cleavage and resealing is what allows the gyrase to actively remove those positive supercoils. It relieves the strain so the helicase can keep moving forward.
It's this beautiful mechanical solution that allows the fork to move at incredible speeds, up to 1,000 nucleotides per second in E. coli.
Speaking of speed, let's look at the drivers of synthesis, the DNA polymerases. The history here is a little confusing.
Yeah, because the first one found, DNA Polymerase I, the Kornberg enzyme, turned out not to be the main replicator.
It's abundant, but it's mostly for repair and cleanup.
The real workhorse is DNA Polymerase III. There are only about 10 copies of this massive pole-free holoenzyme per cell, but it's the major replicative enzyme.
And regardless of which one we're talking about, all DNA polymerases share 2 fundamental requirements. First, they need a template.
They have to have something to copy.
And second, and this is critical, they need a primer, an existing strand of nucleic acid that provides a free 3' hydroxyl group.
And this is the key limitation. Polymerases cannot initiate synthesis de novo. They can't start a new chain from scratch. They can only extend an existing one.
And their directionality is absolute. They all synthesize DNA in only one direction.
The 5' to 3' direction. And the chemistry behind that is fixed. The 3' OH group of the existing strand acts as a nucleophile. It attacks the 5' phosphate of the incoming nucleotide, linking it to the chain.
So you can only add to that 3' end.
Only to the 3' end. And that fundamental chemical reality, combined with the fact that the DNA helix is anti-parallel, is what forces this process of semi-discontinuous synthesis.
Okay, let's break that down. The 2 strands of the template run in opposite directions.
Right. So when the fork opens up, One template strand is running 3' to 5' relative to the fork's movement. The other is running 5' to 3'.
The one synthesized on that 3' to 5' template is called the leading strand.
And that's the easy one. Its synthesis can just proceed continuously, 5' to 3', right toward the fork, extending smoothly as the DNA unwinds.
But the other strand, the lagging strand, is the problem. It has to be synthesized discontinuously.
In chunks. Because the polymerase has to move 5' to 3', which is away from the direction the fork is moving.
So it's like it's stitching backwards?
Exactly. The synthesis of each chunk has to wait until the fork is moved forward enough to expose a new section of template. The evidence for these chunks, the Okazaki fragments, came from experiments showing that new DNA initially exists as these short segments.
Which are then ultimately joined together by DNA ligase.
Right. But before that can happen, we have to solve the priming problem. If Pol III can't start a chain, how does each of those Okazaki fragments even begin?
You need a different enzyme.
You call in primase. It's a specialized RNA polymerase. And primase can initiate synthesis de novo. It lays down a short RNA primer, about 10 nucleotides long, on the lagging strand template.
And then DNA Pol III recognizes the 3' OH on that RNA primer and takes over from there.
And synthesizes the DNA part of the Okazaki fragment. The cell basically accepts using RNA, a less stable molecule, to get started because those RNA primers are easy to identify and remove later. It prevents initiation mistakes from being permanently baked into the DNA.
Okay, so let's assemble the whole machine at the fork, the replisome. We start with the enzyme that pries the helix apart.
DNA helicase. It's this remarkable 6-subunit ring that actually encircles the lagging strand template. It uses ATP to just power its way down the strand, breaking the hydrogen bonds.
And once the strands are separated, they're fragile.
Very. So single-stranded DNA-binding proteins, or SSBs, immediately coat the exposed single strands to prevent them from snapping back together or getting damaged.
And the helicase and primase work together in a unit called the primosome.
Right. But the real mechanical masterpiece is how the two Pol III core enzymes, one for the leading strand, one for the lagging, are coordinated. This is the trombone model.
The whole replication complex is physically tethered together.
Yes. But the lagging strand polymerase has to synthesize in the opposite direction of the fork's movement. To solve this, the lagging strand template actually loops back on itself.
So it's fed through the complex, forming an ever-growing loop of single-stranded DNA.
Exactly. The loop grows and grows until the Pol III core finishes that Okazaki fragment. When it hits the primer of the previous fragment, it lets go, the loop collapses, and the polymerase immediately cycles to the next RNA primer closer to the fork.
And the loop reforms, like the slide of a trombone.
That's the idea. It allows the entire replisome, both polymerases, to move together in the same linear direction down the DNA.
The source also mentions a modern refinement, that there might be 3 copies of Pol IV.
Yeah, the idea is that having 2 dedicated to the lagging strand allows for more seamless handoffs. As one finishes a fragment, the other can immediately start the next one. It minimizes any waiting time, which is critical for maintaining that incredible speed.
That mechanical description leads us right into the complexity of the polymerases themselves. The DNA polymerase III holonym isn't just a synthetic enzyme. It's a factory built for speed and endurance.
And it has to solve what we call the processivity paradox.
Okay, what's processivity?
It's the ability of a polymerase to stay tightly bound to the DNA and synthesize a long stretch of nucleotides without falling off. For replication to be fast, the polymerase needs to be extremely processive.
But if it's bound that tightly, you'd think it would slow it down. It would be hard to slide along the DNA.
And the solution is an engineering marvel, the beta sliding clamp. This protein is literally a donut-shaped ring that completely encircles the DNA duplex.
So the polymerase itself isn't touching the DNA directly.
The core polymerase is tethered to the clamp. The clamp can slide freely along the DNA with almost no friction, but it keeps the polymerase from ever floating away from the template. It's like a lanyard.
So how does this donut-shaped protein get onto the DNA helix in the first place? It can't just slide on from the end of a circular chromosome.
It needs help. From the multi-subunit clamp loading complex, this is another key part of the holoenzyme. It uses the energy of ATP to bind the beta clamp and literally crack it open.
It tries the ring open.
Into a spiral shape, yeah. It threads the DNA through the opening and then allows the clamp to snap shut around the DNA. Once the clamp is on, the loader dissociates and the polymerase can dock and start synthesizing.
Which explains why the lagging strand needs a whole new clamp loading operation for every single Okazaki fragment.
It really underscores the complexity. And while pol III is the workhorse, let's go back to polymer for a second. Its main job is repair, but it's essential for finishing replication because of one of its other activities.
It's a 3-in-1 enzyme. It has the polymerase activity, the proofreading.
And the 5' to 3' exonuclease activity, and that's the one that's critical for cleanup.
What does it do?
It moves in the direction of synthesis, chewing away the RNA primer ribonucleotides from the 5' end of the Okazaki fragment ahead of it. At the same time, its polymerase domain fills the gap behind it with DNA.
A process called Nick translation.
Right. And once the whole primer is gone and the gap is filled, DNA ligase comes in and seals that final nick, making the strand whole.
Okay, let's talk fidelity. The error rate is astonishingly low, less than 1 in a billion. How?
It's a 3-layer defense. Layer 1 is just accurate nucleotide selection.
This is about the shape of the active site.
Exactly. The polymerase is designed to accept only the geometrically perfect base pair, AT or GC. They have virtually identical shapes. If a mismatch tries to get in, the size and shape are just wrong.
So it's a physical filter. But then there's a second layer, induced fit.
This is the chemical checkpoint. The polymerase doesn't just wait passively. Only if the incoming nucleotide correctly base pairs with the template can the fingers domain of the polymerase close around it. If the geometry is wrong, that structural shift, the induced fit, fails. The reaction can't happen.
But even with those 2 checks, you still get errors, maybe 1 in 100,000 bases or so.
Which is still way too high. So that brings us to the third and most powerful layer, immediate proofreading.
The 3' to 5' exonuclease activity.
Right. If an incorrect base does get incorporated, the mismatch subtly distorts the helix. The polymerase stalls. It feels that distortion.
And then what happens?
That stalling causes the newly synthesized 3' end to fray a little bit, and it gets physically shifted out of the polymerase active site and into a separate exonuclease site on the enzyme.
It's like hitting the backspace key.
A mandatory rewind. The exonuclease chews back and removes the wrong base, and then the corrected 3' end is put back into the polymerase active site to try again. This step alone removes about 99 out of every 100 mistakes.
Pushing the error rate way down.
And then a final system, post-replicative mismatch repair, cleans up most of what's left, getting us to that final, incredible fidelity.
Let's shift gears and look at the dark side of replication without proofreading. Viruses, specifically HIV and its reverse transcriptase.
Right. RT uses an RNA template to make DNA, but the key thing is it lacks that critical 3' to 5' proofreading exonuclease, so its error rate is much, much higher.
Which is why RNA viruses evolve so quickly.
Exactly. But that high error rate is also its Achilles heel, and medicine has exploited it. The drug AZT is a perfect example.
Azithromycin.
It's a nucleotide analog that looks like thymidine. Because RT is so sloppy and error-prone, it frequently grabs AZT and incorporates it into the growing DNA strand.
But AZT has a trick.
It crucially lacks the 3' OH group.
Yeah.
So once it's incorporated, the chain is dead. Nothing else can be added. It's a chain terminator, and it selectively halts viral replication.
Before we leave this area, the source touches on this amazing idea of storing digital data in DNA.
It's a compelling concept. DNA is incredibly stable, it can last for thousands of years, and it's unbelievably dense.
So researchers have encoded images, text, all sorts of things into synthetic DNA. What's the catch? Why aren't we all using DNA hard drives?
Cost and speed. Reading and writing data is currently very slow, taking hours, so it's only good for long-term archival storage. But the other problem is that the synthesis process itself is imperfect.
Wait, but you just said polymerases are nearly perfect?
Biological polymerases are. But this is synthetic chemistry in a lab, not biological replication. And because the synthesis is error-prone, the engineers have to build in all sorts of sophisticated error correction codes, which means you can't even use the full theoretical storage capacity of the DNA.
That brings us to the unique challenges of eukaryotic replication. We're dealing with a genome that's maybe 1,000 times larger, packed into chromatin, and our polymerases are actually slower than bacterial ones.
Scale is the #1 problem. If a human genome only had one origin of replication, it would take weeks to copy everything. But it all has to get done in the few hours of S phase.
So the strategy has to be massive parallelization.
Exactly. The genome is divided into hundreds of thousands of small, independently regulated units called replicons. In humans, replication starts at maybe 10,000 to 100,000 different origins all across the genome.
And the timing of when these origins fire isn't random.
Not at all. It's highly regulated and correlates directly with gene activity. Euchromatin, the open, active regions of the genome, tends to replicate early in S phase.
And heterochromatin, the tightly packed silent stuff, replicates much later.
Right, and this coordination likely helps to preserve the patterns of gene expression when the cell divides.
But having so many origins creates a new problem. How do you make sure that each one fires once and only once per cell cycle? Copying a region twice would be catastrophic.
This is handled by a beautiful process called replication licensing. It's a three-step process tied to the cell cycle. Step one is origin recognition.
This involves the origin recognition complex, or ORC.
ORC binds to the origin sequence and just sits there, acting as a landing pad throughout the entire cycle.
Step 2 is the actual licensing.
This happens only during G1, when the activity of an enzyme called CDK is low. The ORC recruits other factors that load the MCM proteins onto the DNA.
And the MCM proteins are the eukaryotic helicase.
They are. Once they're loaded, that whole structure is called a pre-replication complex, or pre-RC. The origin is now licensed, it's competent to fire.
And then step 3 is firing.
At the start of S phase, CDK and another kinase, DDK, become highly active. They phosphorylate the MCM complex, which activates the helicase, and replication begins. But crucially, that now high CDK activity suppresses the formation of any new pre-RCs.
So it's a one-way switch. You can only load the helicases when CDK is low in G1, and you can only fire them when CDK is high in S phase.
And that high CDK level then prevents reloading until the cell has gone all the way through mitosis and CDK activity drops again. It's a perfect molecular timer.
The eukaryotic replication fork itself has components that are similar to the bacterial ones, but the details are different.
Right. The Okazaki fragments are much smaller, around 150 nucleotides, and the polymerases are highly specialized.
Okay, walk us through the 3 main replicative polymerases.
First, you have pol alpha primase. This complex initiates everything. The primase subunit makes the RNA primer, and then pol alpha extends it with about 20 DNA nucleotides. It starts every Okazaki fragment, but it doesn't have proofreading.
Then the main workhorses take over.
Those are pol delta and pol epsilon. They are the highly processive, proofreading-capable enzymes. Pol epsilon is thought to handle the continuous leading strand, and pol delta takes care of the lagging strand.
And both of them depend on the eukaryotic sliding clamp, PCNA.
Absolutely. PCNA is essential. Like the beta clamp, it gives them processivity. But PCNA is often called a molecular tool belt because it does so much more. It recruits a huge number of other factors for repair, for chromatin assembly.
And for primer removal, which is also different in eukaryotes.
It is. Pol delta just keeps synthesizing until it runs into the primer of the previous fragment. Doesn't stop, it just displaces it, creating a little single-stranded flap.
And that flap gets cut off.
By an enzyme called FEN1 endonuclease. It clips it off precisely, and then DNA leagy seals the neck.
And where are all these thousands of forks actually working inside the nucleus?
They're not just scattered randomly. They're localized in discrete zones called replication foci. Each focus might contain a cluster of dozens of active forks, probably coordinating the replication of adjacent regions of a chromosome.
But the biggest challenge that's really unique to eukaryotes is dealing with chromatin. The machinery has to disassemble the nucleosomes ahead of the fork and reassemble them almost instantly behind it.
And the inheritance of the histones themselves is very complicated. The core histone octamer actually breaks apart. The central H3H42 tetramers stay intact and get randomly distributed to the 2 daughter DNA strands.
So a daughter strand gets a mix of old and newly synthesized histones.
Exactly. And this whole reassembly process is mediated by proteins called histone chaperones. One of the key ones, CAF1, is recruited right to the fork by binding to PCNA, reinforcing that tool belt idea.
This leads to the critical question of epigenetic transmission. The cell needs to copy not just the DNA sequence, but also the instruction manual written on top of it.
The pattern of DNA methylation and histone modifications that tell a cell whether it's a skin cell or a neuron.
So how does the cell maintain that pattern?
DNA methylation patterns are partly maintained by an enzyme, DNMT1, that sees the methylation on the parent strand and copies it to the new strand. Histone modifications are thought to be maintained through a positive feedback loop.
How does that work?
Well, if a specific epigenetic mark is present on an old parental nucleosome, that mark acts as a binding site for proteins that then recruit the enzymes that catalyze that same modification on the new nucleosomes nearby.
So the existence of the mark guarantees its own propagation.
It's a way of making sure that a region that was condensed stays condensed, and a region that was open stays open, maintaining that inherited cellular identity across cell divisions.
We started by saying our genomes suffer about 10,000 lesions every day. That brings us to our final and maybe most critical segment, DNA repair.
And the core principle of repair is thankfully consistent. Find the damage, cut it out, and use the other strand as a perfect template to rebuild it.
So let's start with nucleotide excision repair, or NER.
NER handles bulky lesions that really distort the double helix. The classic example is the pyrimidine dimers you get from UV exposure.
And NER operates through two different pathways.
Right. There's the super-efficient transcription-coupled pathway, which prioritizes active genes. The damage is actually signaled when an RNA polymerase physically stalls at the lesion.
And then there's the slower background pathway for the rest of the genome.
The global genomic pathway. But the mechanism of repair is similar. You need a huge complex, including a factor called TFIIH, which has helicase subunits that unwind the DNA around the lesion. Then endonuclease cut the damaged strand on both sides, removing a chunk of about 24 to 32 nucleotides.
And then pol delta or epsilon fills the gap.
And ligase seals the nick.
Okay, next up is base excision repair, BER, for smaller, non-distorting damage.
Like when a cytosine spontaneously deaminates and turns into a uracil, or when bases get oxidized by metabolic byproducts.
And BER is initiated by a very specific type of enzyme.
A DNA glycosylase. There are many different kinds, each one specialized to recognize a specific type of damaged base. And what it does is it cleaves the bond, linking the base to the sugar, removing only the base.
Leaving behind what's called an AP site.
A beheaded sugar. And the way the glycosylase finds the damage is incredible. It scans along the DNA and forces each base to flip 180 degrees completely out of the helix and into its active site for inspection.
So it's an active, physical quality control check.
If the base is damaged, it fits the active site and gets cleaved. If it's normal, it doesn't fit and just gets flipped back into the helix. Once the base is gone, other enzymes come in to remove the rest of the sugar phosphate backbone, and a polymerase fills the one nucleotide gap.
We've mentioned mismatch repair, MMR, as that final layer of fidelity. Its biggest challenge is figuring out which of the two mismatched bases is the wrong one.
It has to know which one was the original template and which was the newly synthesized mistake. In E. coli, it can tell based on methylation patterns. In eukaryotes, it's less clear, but we know that defects in MMR are catastrophic. They lead to incredibly high mutation rates and are strongly linked to certain types of colon cancer.
And that brings us to the most dangerous damage of all, double-strand breaks, caused by things like ionizing radiation.
Literally snapping the chromosome in half. There are two main ways to fix this. The first, and the most common one in our cells, is non-homologous end joining, or NHEJ.
NHEJ is the quick and dirty fix.
It's the cell's emergency system. Proteins like Ku grab the two broken ends to protect them, and then DNA linkage basically just forces them back together. It often involves trimming the ends, so you usually lose a few nucleotides. It's fast, but it's betagenic.
The second, much more accurate method is homologous recombination, HR.
HR is template dependent. It can only happen in the late S or G2 phases of the cell cycle after the DNA has been replicated because it needs to use the identical sister chromatid as a pristine template to ensure perfect repair.
And as we said at the start, BRCA1 is a central player in this high-fidelity HR pathway.
Absolutely.
Let's finish by connecting replication and repair. What happens when a polymerase is chugging along and hits a patch of unrepaired damage?
The main replicative polymerases, pol, delta, and epsilon, just stop dead. They are too precise to handle a damaged template. When that happens, the cell calls in specialized enzymes for translesion synthesis, or TLS.
And these TLS polymerases, like POLT-ETA, are fundamentally different.
They're the cell's skeleton keys. They have no processivity and, crucially, no proofreading. Their active sites are much more spacious and can physically accommodate a damaged base that would stall a normal polymerase.
So they prioritize just getting past the roadblock, even if it means putting in the wrong base.
Exactly. They are error-prone, but that's the trade-off. They synthesize just a few bases to get past the lesion, and then the high-fidelity polymerase is swapped back in to continue.
But the stalling of the replication fork itself is dangerous.
Very. It can lead to fork collapse and genomic rearrangements, like copy number variations, which are implicated in all sorts of disorders.
Let's conclude then with the very real consequences of DNA repair deficiencies, which are highlighted by these distinct human syndromes.
You can really see the difference based on what function is impaired. In xeroderma pigmentosum, or XP, patients have a fundamental defect in nucleotide excision repair.
So they can't fix UV damage.
Which leads to a massive accumulation of mutations and an extremely high risk of fatal skin cancers.
But then you have a syndrome like Cockayne syndrome, CS. They're also sensitive to sunlight, but they typically don't get more cancer.
And this is the crucial distinction. CS patients have a specific defect in the transcription-coupled part of NER. They can't quickly repair actively transcribed genes, which leads to widespread cell dysfunction and programmed cell death, or apoptosis.
So it's a difference between mutation accumulation leading to cancer versus apoptosis leading to features of accelerated aging.
It is. And we even see this with different mutations in the same gene. Different mutations in the XPD gene can cause XP or CS or a third disorder. The idea is that one mutation might cripple the repair function, leading to cancer, while another might cripple the transcription link, leading to apoptosis and aging.
It all highlights the pervasive impact of compromised genomic maintenance on our overall health and survival.
Absolutely.
This has been a huge deep dive covering the absolute molecular foundations of life. We started with the simple, elegant proof of semiconservative replication.
And then move through the incredible machinery of the prokaryotic replisome, the trombone model, the sliding clamp, and that three-tiered defense that ensures near-perfect fidelity.
We then scaled that up to eukaryotes, seeing how they solve the problems of scale with replicons and precise licensing, all while managing the huge challenge of replicating through chromatin and maintaining epigenetic memory.
And finally, we explored those constantly vigilant repair systems,
Yeah.
NER, BER, and the high-stakes choice between the fast but messy NHEJ and the perfect homologous recombination pathway.
It's a system under constant daily assault, yet it achieves an error rate that engineers can only dream of.
So let's end with a provocative thought, one that links repair and inheritance. We saw how carefully the cell manages the distribution of old histones to guide those positive feedback loops for epigenetic maintenance during normal replication.
Right, to preserve cell identity.
But now consider the error-prone, non-homologous end-joining pathway. This repair often involves chewing back the DNA ends, causing deletions, and just violently disrupting the local chromatin structure.
Given that NHJ is active all the time and often results in losing some information, what do you think the lasting consequence is? The ultimate loss of inherited cellular identity in a region of a chromosome that's been salvaged by that pathway.
The physical disruption means the old histone placement, the epigenetic marks, they're gone. Does that region ever truly recover its historical programming? That's something to ponder as you reflect on the constant trade-off between speed and fidelity in keeping our genome intact.
Thank you for joining us for this deep dive into DNA replication and repair.
Until next time.